Okay, I'm not hearing anyone. Can somebody confirm you guys can hear me?
Can hear you.
Yeah. All right, before we begin, are there any questions? I don't hear anything, so I'm assuming there's no questions. Let me share my screen here. All right, you should be seeing my screen now. Today is October 9th, and we should be on chapter 3. I forgot to look up whether the quiz 2 is going to happen this week or next. Next week, I mean, next week or the next. Let me quickly look into that. Myself a note, but then I forgot to actually do that. Let me look here. All right, let me stop the share because I'm going to look at. I'm going to move quiz 2 on the syllabus down to week 3, which it has that in Canvas. One advantage for that is it's going to have chapter 3 on quiz 2, and we're not going to be finished with chapter 2 by next week, at least not until later in the week. Let me move that over. Erased. See if I can truncate that. Yep, can. All right. I'm going to share my screen again. I think I'm out of the gradebook. Let me get out of the gradebook just in case. All right, for those of you who didn't follow what I was saying, I moved quiz 2 down a week in the syllabus. It was correct in the Canvas. Let me look here. What week is that supposed to be? I don't know if I have a link for it on the main page, but so quiz 2 has been moved to week 5, Wednesday, week 5, which it is that on Canvas. Let me make a link here in the main page.
I do have a question with the quizzes.
Just give me a minute here and then I'll be able to answer. I don't even see quiz 2 here. All right, let me add quiz 2 here. All right, what's your question?
Um, I couldn't remember if the syllabus said are for the lowest quiz that has the lowest grade, are you going to be dropping that score?
Yeah, that's in the syllabus. You
Okay.
have um, 300 points on the quizzes, but we're taking five of them. And so one of the quizzes, the points has to be dropped.
Okay, thank you.
Okay. All right. Anyways, that's all. I got quiz 2C in week 5. And my apologies that the syllabus is not correct. I've got to upload that now. Make a note of that. There we go. Hopefully I'll remember that I've got a note there because like I said, with the looking into this, I was supposed to do that after the lab and totally forgot about it. All right. Um, let's move on. Let's go to the syllabus here. So today we're supposed to be working on chapter 3, observing microorganisms. And that's it. For those of you in the lab at 6:30, we'll be doing lab 4. I think I'm done with this. So, well, I'll use it for the lab. I'll keep it up. Someplace I've got that. There we go. So we had talked about the metric system, and there's two reasons why you should know the metric system. First, science and medicine use the metric system. And the other reason is this map is showing you the countries in the world that do not use the metric system, which is very few of them. All right, you should know the typical sizes of microorganisms. A prokaryotic cell ranges, well, the exact range would be something like zero point 0.2 to zero point 0.4 micrometers up to about 10 to 20 micrometers. And this has a range between 1 and 2. The average would be 1 to 2 micrometers. The eukaryotic cell, the average for that one depends on which cell we're talking about, is about 10 times larger. And this range is from about 10 micrometers to 500 micrometers. But remember, the largest eukaryotic cell is the yolk of a chicken egg, and that would be close to something like 2.4 centimeters. So eukaryotic cells vary quite a bit as well. Uh, Viruses are about 10 times smaller than a prokaryotic cell, with the smallest being about 200 nanometers, that's 0.02 micrometers, to the biggest being about 0.44 micrometers, that's 440 nanometers. And you should note that the very largest viruses are actually a little larger than the smallest prokaryotic cell. Viruses can depend on what the shape of the virus is. I gave you the virus diameter. The longest virus is actually Ebola, which reaches 1 micrometer. However, Ebola is very thin, so it is not the biggest virus by diameter. Megavirus is about the biggest virus by diameter. Any question about any of that? You should know that we're going to talk about things like humans, about the biggest thing we're going to talk about. Chicken egg, the biggest eukaryote cell, at least the yolk of the chicken egg is a cell. And then frog eggs, about the smallest cell that you can actually see with your eyes. You can see a little bit smaller, but about frog eggs. And then we have the things that light microscopes can see. That includes most eukaryotic cells, prokaryotic cells, and about the smallest bacteria you can see with the light microscope. Viruses are smaller, generally, and you cannot see them with the light microscope. Obviously, the very, very largest viruses you could see with the light microscope, but most you cannot see with the light microscope, and we need an electron microscope to see viruses. Things smaller than viruses are large biological molecules, like a large protein or a large carbohydrate, a large lipid. Small molecules are then underneath a large molecule, and then atoms are smaller than a small molecule. Small molecule would be something like water. Any question about any of that? Note that there is a mistake in this graph. It's got a micrometer in there twice, once here and once there. I've made a correction there. That one there should be a millimeter, about the size of a frog egg. You should know all the parts of the microscope shown here. The microscope that we have in the lab are compound light microscopes. A compound microscope is one using two or more lenses that magnify. And we have a objective lens here and an ocular lens there that magnify. The total magnification is the magnification of the objective lens times the magnification of the ocular lens. Typical microscopes, like almost all of them in the microbiology lab, have four objective lenses: the 4x lens, the smallest, the 10x lens, the 40x lens, and the 100x lens. I sometimes call the 100x lens the oil immersion lens and the 4x lens the scanning lens. I do not call the 10x lens the low power lens because it's not the lowest, but you can, that is the name of it. The 10x lens is called the low power lens. And then the 40x lens, I don't use that name either. It's called the 40x lens, it's called the high power lens, but it's not the highest. The 100x lens is. So if you use the long names, you should remember that 10x is not the lowest and the 40x lens is not the highest. It is the low power lens and the high power lens. And I don't use those terms because students frequently get them confused and mixed up. So you should know the parts of the microscope detailed here. The ocular lens. Don't really need to know the path of the light, but the light comes from the lamp, going through the specimen, going through the specimen, and then going into the objective lens, being collected by the objective lens, and then focused on the eye. You should know the body tube, that's what holds the microscope together. The arm, which isn't here, labeled here, but that's what you carry the microscope by. You should know the condenser lens, making a cone of light shining on the specimen. The specimen where we have the stage and the slide. We don't have it shown here, but the iris diaphragm is with the condenser. The illuminator is the lamp, and then the base for the microscope. Our ocular lenses are always 10x, but the most expensive microscopes have a 20x ocular lens. The ocular lens can vary in power. Like on a child's microscope, I've seen the ocular lens 4x time, but most microscopes have an ocular lens of 10x, including, as far as I know, all of the microscopes at Clark College. And yes, we do not have the most expensive microscopes. So here are the parts of the microscope. I already talked about the first three. The objective lenses, we've already talked about those. The stage to hold the microscope. Stage clip, which you can see, it's not labeled here. The condenser. On some microscopes, the condenser is above the iris diaphragm. On some microscopes, the iris diaphragm is above the condenser. The iris diaphragm opens and shuts like a shutter And what you want to do with the iris diaphragm is make the cone of light equal to the field of view, and then you'll get polar illumination, the best illumination you can from that lens. The illuminator, the light source, the base of the microscope, and then the fine focusing, the small knob, and then the coarse focusing, the large knob. Any question about any of that? This is a compound microscope because it has two microscopes that magnify, excuse me, two lenses that magnify the objective lens and the ocular lens. This is a simple light microscope because it only has one lens that magnifies. And a simple microscope is nothing other than a very fancy magnifying lens that also magnifies a lot. This microscope was actually made by Antony van Leeuwenhoek. And he was one of the first microbiologists, certainly the first bacteriologist. The resolution of a microscope is the ability of the lenses to distinguish 2 points as two separate independent points. If they get too close together, you cannot distinguish them as two independent points. With microscopes, the resolving power is about 0.2 micrometers, which is the closest 2 points on a microscope can be, and then you can still see them as two independent points. The reason for this limit is that it is the limit, the resolution limit of white light, about 0.2 micrometers. That's 200 nanometers. Anything closer together, white light cannot distinguish them as two separate independent points. And then, of course, a light microscope cannot distinguish them as two separate independent points. The best compound light microscope can magnify 2,000 times. That would be a 100x objective lens and a 20x ocular lens. Anything or any magnification beyond this power is empty magnification, because if you're magnifying at this power 2,000 times, you're getting down to 0.02 micrometers, and then you cannot, even though you have extra magnification, it's not doing anything because you cannot magnify anything smaller than 0.02 micrometers. Is that clear to everyone? So the resolution limit of white light is 0.2 micrometers. And we get that when we're using a total magnification of 2,000 power. So if anyone were to ever make a microscope that had higher magnification, it's empty magnification because you're not seeing any additional detail, meaning 2 points that are closer than 0.2 micrometers together, you would not be able to see them as two separate independent points. And here's a good picture to show you. These 2 points shown here in real life, that's with the light microscope, you can separate these as two independent points. These right here, you cannot separate as two independent points or independent points. And that's because they're closer together than 200 nanometers or 0.2 micrometers. Any question about any of that? And the reason why you can see these as two independent points and then all of these you cannot is because these are 200 nanometers apart and white light can resolve them as two independent points. These are less than 200 nanometers apart and white light cannot resolve them as independent points. And it has to do with the wavelength of the light. Why I said white light. You can only resolve things down to about the wavelength of light. I think specifically it's half the wavelength of light is the resolution limit. All right, any question about resolution? So with greater resolution, you have greater clarity of the object under the microscope. Light undergoes refraction. The refractive index is the light bending ability of a medium, the substance through which light passes. When we see this pencil here, it looks like the pencil is bent or broken right there. It isn't, it's a straight pencil, but the reason why it looks like the pencil is curved is because the light is refracting differently in water than it is in light. And at the interface, the light will bend, meaning the interface between the water and the air, the light will bend, and that we call the refractive index, the amount that the light bends. And it's because of the light bending at this water to air interface that it makes the pencil look like it's bending right there. Any question about any of that? And there's the artist's rendition of the pencil bending. It's not actually broken. It's just bending because the light is refracting at the water to air interface. And the reason why the light is bending there is because the water has a different refractive index than air. And so when the light changes media, some of that light is bent. Any question about any of that? It is because of the refractive index of light that we use oil on a microscope. Oil has the same refractive index as glass. And so when you put oil between the glass slide and the glass of the objective lens, you have the light going from glass to oil to glass, and the light will never refract. Any question about any of that? Refraction is actually very important for life. Like when a when a bird sees a fish in the water and the bird wants to catch the fish, because of the refraction of light, the fish is actually down in this position, but it will look to the bird like the fish is up further in this position. And the reason for this difference is because of the refraction of light at the water to air interface. Birds instinctively know that when they see the fish here, the fish is actually in this position because of the refraction of light, and then the bird will put out its feet to catch the fish in this location, not in this location. Any question about any of that? And this is very important for life because if birds didn't compensate for the refraction of light, they would have a much harder time of catching the fish. Any question about any of that? All right. So the reason why we put oil between the slide and the objective lens is that when we have oil there, the light will not be refracted from the glass to the oil or from the oil to the glass of the objective lens. So the light will travel straight from the specimen through the oil to the objective lens. If, on the other hand, we have air in between the glass of the slide and the glass of the objective lens, the light rays coming out of the glass, some of them will refract, and some of them will refract at such a strong angle that they will not be collected by the objective lens. The light will also refract from the air to the glass of the objective lens. So we'll have refraction in two places. Any question about any of that? What does that mean? It means when you have light coming through the slide and going through the specimen, if you use oil, more of the light will be collected by the objective lens than if you don't use oil, because this ray is not going to be collected by the objective lens just because it's at such a strong angle. It will not be collected by the objective lens, but this one and that one will be collected as well as that one. And that one's traveling straight from the lamp through the specimen to the objective lens. These are at an angle, but they will be collected. And then if you don't have oil, the light will be refracted. At least some of the light will be refracted. Some of the light will travel through the slide, go straight, will not be refracted, and then go straight through the air, going straight through the objective lens. Okay? Other light rays will be refracted in the slide to air interface, but will be collected by the objective lens. And some of those will be refracted, and that's why it's bending again right there. But some of the light rays being refracted out of the glass will be refracted at such a steep angle that some of those light rays will not be collected by the objective lens. And then some of the light being refracted out of the glass and into the air interface will actually be refracted so hard that the light will be sent back into the glass and go through the slide someplace over here. Okay? And that would be something like this one there. The point being is that more light rays will be collected if we're using oil, so the objective lens will gather more light than if you don't use oil, where some of the light will be refracted away from the objective lens. And because we're collecting more light with oil, you'll have greater clarity and greater resolution because you're getting more light than if you don't use oil because you'll get less light rays. Any question about that? And obviously light will have different rays. Some of it will not be refracted even when going through glass, air, and then glass. Some of it will be collected, but will be refracted at the glass to air interface, as well as the air to glass of the objective lens interface. The reason why the light ray does not refract or bend in the case of oil is because the oil has the same refractive index as glass. And that's why we use immersion oil, to get greater collection of light rays and to get a better resolution. Any question about any of that? All right. So bright field illumination is what we normally see with most light microscopes. All of the compound light microscopes we have in the microbiology lab use bright field illumination. In bright field illumination, you have light rays coming from a lamp or light source, going through a condenser lens, going through the specimen and a cone of light from the condenser lens, and then being collected by the objective lens, coming to the eye. The specimen will look bright in the background because all we're seeing there is light going through the glass slide, and the glass is transparent. So it'll be a very bright background. The object will be darker but bright, also bright. compared to this one here. We'll talk about that in a minute. So this is why it's called bright field illumination, because the background is very, very bright. All you see really is the light. And then the specimen is also bright, although darker than the background, obviously. And bright field illumination is most compound light microscopes, including all of them we have in the microbiology lab. This contrasts with dark field illumination, which has light coming from a lamp source, but then we have an opaque disk blocking the light rays directly coming from the lamp and then going to the specimen. Because all of the light coming directly from the lamp is blocked, the background is dark, and why it's called dark field illumination. Some of the light rays are coming from the lamp at a steep angle, and when they hit the specimen, they're actually reflected or refracted into what the objective lens can collect, meaning it's only because they're going through the specimen that the light ray will then be changed and then be allowed to be collected from the objective lens. Is that clear to everyone? Because the light rays directly coming from the lamp are blocked by the opaque disc. The only light rays that can hit the specimen are something coming at a very strong angle, and this light ray would not be collected by the objective lens unless the light were to be bent, and then it can be collected by the objective lens. That makes the background very dark because all of the light from the lamp source is being blocked by the opaque disc, but some of the light coming from the specimen will be bent in an angle so that the objective lens can collect it, and that makes the object here darker than the object in bright field illumination. And then the background is much, much darker. It's totally black in dark field illumination. Whereas in bright field illumination, the background is totally light. Any questions about bright field illumination compared to dark field illumination? You don't need to know dark field illumination. I need a K there. Other than the concept of dark field illumination, you don't need to know specifically what's going on in dark field illumination, but you should know that in dark field illumination, the background is dark and the specimen is darker than in bright field illumination. The reason why is because light rays directly coming from the lamp are blocked by an opaque disc. All right, any question about any of that?
Is there a reason you would use one over the other?
That's a good question. Let me think about that. If you want to see the background of the cell, you need to use bright field illumination. So if you're using like a negative stain or you're looking for a cell capsule, you need to use bright field illumination. On the other hand, if you don't care about the background of the slide and all you want to see is the specimen, you could use dark field illumination, although you can see the specimen under bright field illumination. So I'm trying to think, why would you use dark field illumination over bright field illumination? I would say the contrast between the specimen and the background of the slide is greater with dark field illumination than with bright field illumination. So if you want to increase the contrast, you can use dark field illumination. And I'm sure there are other reasons why you'd use dark field illumination, but off the top of my head, I just don't know. And I did mention two reasons. One reason why you'd use bright field illumination, and then a reason why you'd use dark field illumination if you wanted the contrast between this cell to be greater to the background of the slide. Any question about any of that? You can read about these in the book, and there's actually other illuminations, which I'm not going to talk about. What is that one called? Phase contrast illumination. It's just too complex to talk about, so I'm not going to talk about it and not quiz you on it. One reason for using phase contrast illumination, which is a little bit similar to dark field, but it's different, is it allows you to see a specimen even though the specimen has not been stained. And we're not going to talk about why that's the case, but it uses several, what are those things called? Never mind. I don't remember the rings. There we go. Ring-like structures in the microscope. And we're not going to talk about it. It's just too complex. Bright field is complex enough. Any question about bright field illumination versus dark field illumination? Other than one I already answered. All right, let's move on to an electron microscope. You can notice something different about an electron microscope from this picture here compared to a light microscope, which, well, there's one there, but let's go there. And that is, you obviously can carry this, so it's much smaller than a electron microscope. An electron microscope is much bigger. So that's the very first difference. And then they use a different source for viewing the specimen. An electron microscope doesn't use a beam of light, it uses a beam of electrons. The beam of electrons is focused with an electromagnetic lens, not with a glass lens. The wavelength of electrons is shorter. It's actually about 1,000 times smaller than the wavelength of visible light. And this will give greater resolution, meaning you can separate 2 points down to a smaller limit than you would with light microscopy. White light, I guess. Not light microscopy, a light microscope. So an electron microscope can allow you to separate 2 points and you'll get greater resolution. Electron microscope allows you examination of viruses, the internal cell structures, which are too small to be seen with a light microscope. Any question about any of that? There are 2 main types of an electron microscope. Now, I'm talking about 2 main types that you need to know. There are actually 4 types of electron microscope, but only 2 of them you need to know. There's a transmission electron microscope shown in the picture on the left, and a scanning electron microscope shown on the picture on the right. Let me blow this up. And then I'm going to ask you, what are the differences between a transmission electron microscope shown here and a scanning electron microscope shown here? What are some differences?
It looks like the transmission is seeing more of the internal, and the um, scanning one is more of the outside of the cell.
Yeah, the scanning electron microscope allows you to see the surface of the cell or the outside of the cell. And there's another difference. What is the surface of this cell? Can you see the bottom of the cell and the top of the cell and the side of the cell? And on this cell, which you're seeing the inside, can you see the top layer or the top layer of the cell as well as the bottom layer of the cell? Not hearing anything? So let me go ahead and tell you, the transmission electron microscope allows you to see one plane through the cell. And you're only seeing the object in 2 dimensions. Is that clear here? This object is only showing you in 2 dimensions. Whereas this object here, we're seeing the object in 3 dimensions. This is the top of the cell, that's the bottom of the cell, and this area here is the side of the cell. And we can see all of them. So the scanning electron microscope allows you to see the cell surface in 3 dimensions, or the surface of some object in 3 dimensions. All right, any other questions about this? Like I said, there are other types of electron microscopes. We'll talk about them at the end. You need to know a transmission electron microscope and a scanning electron microscope. And these images, by the way, are taken by an electron microscope. TEM stands for transmission electron microscope, SEM for scanning electron microscope. Here we're looking at crystal with the scanning electron microscope. And you can see this crystal in 3D. And this is a section through an organelle of a cell. I think this is the chloroplast, if I can remember correctly. And these are the membranes you can see in the chloroplast, meaning membranes of the chlorophyll in the chloroplast. And this is taken by transmission electron microscope, which you can only see the specimen in 2 dimensions. Any question about any of that?
Yeah, I had a question about the electromagnetic lenses.
Uh-huh.
So it's not glass.
The
Is it
lens is not glass on a electron microscope.
so
You
is it
focus the rays of electrons using an electromagnetic magnet.
oh, okay. So there's not even any lenses on there. It's just open with a beam of electrons looking
Well,
through.
you are focusing the electrons, but you're using essentially a magnet to focus those electrons.
Okay, thank you.
But good question. All right. This is a third type of electron microscope. It's called the scanning tunneling microscope. It uses a metal probe to scan a specimen. The resolution you can get down to is 1/100th the size of an atom. This is showing you a protein from E. coli. The scanning tunneling microscopy, you will not be examined on it, but this is the greatest magnification we can get with an electron microscope. 1/100th the size of an atom. Any question about any of that? The fourth type of electron microscope is called an atomic force microscope, and you're not going to be examined on it. It uses a metal and a diamond probe, which are inserted into the specimen. It produces a 3D image. Here is a toxin from Clostridium perfringens that you're seeing with atomic force microscopy. Any question about the electron microscope and the 4 types of electron microscope? As I stated, only the first 2 types I will quiz you on. The third and the fourth types I will not quiz you on.
Is the probe kind of like a camera? Like inserted into the cell?
It's not a camera inserted into the cell. It's a piece of diamond as well as a piece of metal that is inserted into the specimen. And the diamond and the metal are inserted into the specimen so the electron microscope can then see the specimen. So it's not really a camera. It's something you're adding to the specimen in order to see it. much like we add a dye to our cells in a light microscope so that we can see the specimen better. So the metal and diamond probe are added, and I'm not sure how they do that, because if it's a very small object, how do you add a metal? It must be just a few molecules of metal they're adding to each object. And then the diamond, I have no idea how they add that. They must just add very, very small pieces, molecules of the diamond to it. Okay. Like I said, I don't know how they do the diamond one, but it must be just a very small molecules, very small amount of molecules added to the specimen. All right. So let's go back to the light microscope. Here is a preparation. Two preparations of specimens for the light microscope. Which of the cells are easier to view? The cells on the left or the cells on the right?
The right.
On the right.
Yeah, the cells on the right. The only difference between these cells and those cells is that these cells have been stained with a dye. The stain, all it does is increase the contrast between the background of the slide and the cell. And that's why this cell is much more visible, because the contrast between this cell is much greater to the background than the contrast between, I don't know, that cell and the background. So cells are basically transparent, and they don't have much contrast with the background of the slide when they're not stained. And that's why we stain them. And all it does is increase the contrast between the specimen or the cell we're viewing and the background of the slide. All right, with bright field microscopes, most microbes are colorless and have little contrast with the slide or the medium. To see them, we really need to add a dye. Now, there are some exceptions, like if you have a photosynthetic cell, it will have green pigment in it if it's something the pigment is chlorophyll. And then you can see the cell. And that is true for the photosynthetic microbes. But most microbes are not pigmented, and you would have a hard time seeing them. I've never looked at Serratia marcescens, which is pigmented, or Micrococcus luteus, which is pigmented. And I wonder if it's easier to see those cells. It probably is, if you don't stain them. But most cells are colorless and have little contrast with the slide, and so we use a stain to increase the contrast, and then we can make out the cell much better against the background of the slide. And that's all a stain does. It increases the contrast between the cell and the background of the slide. Staining is adding a color to the specimen or adding color to the background of the slide. And this will increase the contrast between the specimen and the background of the slide. Before we do a stain, remember we make a bacterial smear, and this is a thin film of bacteria put in solution on a slide. You allow that water or solution to air dry, you then heat fix it, and then that's it for the bacterial smear. We then can take that bacterial smear and then stain it. So prior to staining microbes, we usually make a bacterial smear, and we usually heat fix the cells as well as air dry the cells. And those two procedures allow the bacteria to stick to the glass. If you did not heat fix the bacteria to the glass, most of the cells, if not all the cells, would wash off the slide in the staining procedure. One advantage to heat fixing is it tends to also kill many species of bacteria, like E. coli. If you heat fix the cells, the E. coli, at least most of them, will be killed. That doesn't kill all species, though. For example, any species that has an endospore, if you heat fix the cell, it may kill the parent cell of the vegetative cell, but it will not kill the endospores. So heat fixing tends to kill the cells, but it doesn't kill all species of cells. Any question about any of that? All right, a stain, also called a dye, is a salt consisting of a positive ion and a negative ion. In a dye, one of the ions is colored. We call this the chromophore. If the chromophore is in the positive part of the salt, or the positive part of the molecule, we then call the dye a basic dye. If, on the other hand, the chromophore, or the colored part of the molecule, is on the negative part of the ion, we then call this dye an acidic dye. Any question about any of that? So that's the difference between a basic dye and acidic dye, and that is in the dye, if the colored portion is on the positive portion, or the cation, of that dye, then it's a basic dye. If, on the other hand, the colored portion is on the negative portion of that salt, or the anion portion, we call it an acidic dye. Bacteria have a slightly negative charge. Therefore, basic dyes are better for staining the cell, because a molecule that has a positive charge, which is a basic dye, will attach to the negatively charged cell, staining the cell. Some examples of basic dyes are crystal violet, methylene blue, malachite green, and safranin. So most of the dyes we use in microbiology are basic dyes. Not all of them, but most of them. Any question about any of that? All right. An acidic dye typically stains the background of the slide and not the microbe. So we're staining the glass of the slide, or the liquid in the slide. We're not staining the cell or the microbes. Some examples of an acidic dye are eosin, acid fuchsin, and nigrosin. Because the acidic dye stains the background instead of the cell, we call it negative staining. This goes back to the old Polaroid films, which, when you developed your pictures, they would give you a negative, and the negative would be the opposite lighting or intensity of the lighting on the photograph, meaning an area that in the regular photograph is lighter, like somebody's nose being hit by the rays of the sun, it's lighter. It will appear darker on the negative Polaroid. And then somebody's parts who are darker on the regular photograph, like the colored portion of your eye is going to be darker than the whites of your eye. And so that on the negative would be lighter than the white part of the eye. And that's why we call it negative staining, because it's sort of the negative of what it really is. And like I said, the term comes from a Polaroid, which, have any of you even seen a negative on an old Polaroid film? Nobody's
Yes.
answering? Oh, yes, you have. All right, good. Because I know that technology, I think it went out almost 20 years ago. So probably not many of you even have seen a negative. But in the old days with the old Polaroid film, when you took your picture, you then take the film to a camera place or film place to develop it, and they develop the pictures and then give you a negative. And with the negative, you could get more pictures developed. That's why they gave you the negative. The reason why acidic dyes stain the background of the cell and not the cell is because an acidic dye has the colored portion of the dye a negative charge, and that will be repulsed from the negative cell. And that's why the background stains and the cell typically does not. Here is two pictures of cells, one with a basic stain. So we use a basic stain here, and this one with a negative stain. Both of the pictures do show you the cells, and you can easily distinguish the cell from the background because of the contrast between the cell and the background. But the basic stain stains the cell, and the negative stain stains the background of the slide, not the cell. Any question about a simple stain? Remember, a simple stain is just using one dye to either stain the cell or the background of the cells. See if there's anything else I want to talk about there. Nope. All right. So staining microbial smears, we usually start with a microbial smear and then we stain the cell, or cells, I should say. There are three different types of stains: a simple stain, a differential stain, and a special stain. A simple stain is either an aqueous or alcohol solution of a single dye. So in a simple stain, you're only using one dye. The primary purpose of this simple stain is to increase the contrast between the background of the slide and the specimen. And that will make the specimen show up and be more observable to an individual looking at the specimen. The stain is applied to a fixed bacterial smear for a certain length of time, and then it's usually washed off, air dried. Well, actually, just dried and then examined. Sorry, we've already made the bacterial smear, so the stain is just washed off and dried. It's usually dyed by just blotting, and then it's examined. All the stains will stain the same when we're doing a single dye. Like if we're staining the cells with crystal violet, all the cells will look purple if we're using crystal violet as a single simple stain. Any question about any of that? A mordant may be used with the stain. A mordant intensifies the stain. It does not color the cell itself, but it binds to and intensifies the primary stain. The functions of a mordant are: it's not a stain, it increases the affinity between the stain and the specimen, it may coat the specimen to enhance it, thereby making it easier to see it. Like some of the flagella stains, what they do is they coat the flagella with many layers until the flagella becomes visible. And the mordant is what's coating the flagella, at least in this example of a flagella stain, which we're talking it here as a simple stain. A flagella stain could also be a special stain because we're looking specifically at the flagella of the cells. When we mentioned in the gram stain that, which I guess we only mentioned it for those of you who had the first lab, but we'll talk about it in the second lab. But we have a mordant in the gram stain, and it is grams iodine. Iodine is not being used as a stain here, but that doesn't mean that iodine cannot be used as a stain. Like iodine can be used to stain cells that store or make starch. And then the iodine will color the region of the cell where there is starch. My point is that when we use iodine to color the starch in the cell, we're using it as a simple stain, not as a mordant. But when we use grams iodine in the gram stain, it is used as a mordant. The simple stain would be if you're only using like crystal violet. It's not even the gram stain, it's just a simple stain. Any question about any of that? Hopefully I didn't confuse you on that. All right, let's talk a little bit about a differential stain. Differential stain usually uses two stains or dyes, but that is not required for a differential stain. The differential stain reacts differently with different kinds of microbes, and therefore you can visually distinguish between the two types of microbes. The most commonly used differential stains are the gram stain and the acid-fast stain. If we use the gram stain, it will distinguish between two types of microbes, the gram-positive cells and the gram-negative cell. And that's why the gram stain is a differential stain. The gram stain actually does use two dyes, but like I said, the differential stain does not require two dyes, but usually it does have two dyes. And that's to stain both types of cells. And that's why we have two dyes. All right, any question about differential stains? If not, let's talk briefly about the gram stain. You don't need to know when it was invented, but you should know it was invented by Hans Christian Gram. He was a doctor who developed the gram stain to differentiate between the bacteria causing infection and then the patient's own cells. And what he did was you use the gram stain to stain the bacteria, which were staining purple, and then he saw the patient's cells staining pinkish or gram negatively. And that's the way he used the gram stain. Other microbiologists started using the gram stain, and they quickly realized that the gram stain separated bacteria into two types of cells. The gram-positive cells, which stain purple, and then the gram-negative cells, which stain pinkish. The difference is due to differences in the cell wall, and we'll talk about that later. Let me go through the gram stain procedure in brief. First, you make a bacterial smear on a glass slide. You need to have a bacterial smear in order to do the gram stain. Remember, the bacterial smear is not a part of the gram stain. It is not a part of any staining procedure. It is its own procedure to make the bacterial smear. I'm not going through how to make a bacterial smear, but once you have the bacterial smear, you need to let it air dry, and then you need to heat fix it. You need the air drying and the heat fixing to stick the bacteria cells onto the glass of the slide. Otherwise, the staining procedure will remove these cells from the slide, and you'll remove most of them if not all of them, depending on how many cells you put on the slide. But you will remove most of them if you don't air dry and heat fix. Then you flood the slide with crystal violet, applying it for 1 minute. Crystal violet is the primary stain, and that should be right there. That's not in there. It's the primary stain because it's the first stain we're using. You then wash, dump the crystal violet off after 1 minute, wash with cold water, and then flood, wash, get the water off, then flood with iodine. Iodine is a mordant here. You apply the mordant for 1 minute. Its purpose is to complex with the crystal violet to make a larger complex, the crystal violet iodine complex. You then wash, dump the mordant off, wash with cold water, dump the water off, and then flood the cell with the decolorizing agent. In this case, we're going to use a decolorizing agent of acetone ethanol. And the best way to apply it, it does say to apply it for about 5 seconds, but a better way to apply it is to hold the slide up to your face so you can view when you're applying the acetone alcohol mixture. And once the decolorizing agent stops streaking the blue, stop applying the decolorizing agent. And this will be about 5 seconds. You don't want to go much longer than 5 seconds because the decolorizing agent will eventually pull the color out of the gram-positive cells. Any question about any of that? All right. The decolorizing agent will pull the crystal violet iodine complex out of the gram-negative cells, assuming that it's applied correctly. And then you wash immediately the slide. Why immediate? Because you don't want to leave the decolorizing agent on for any longer length of time. Even if you dump it off, there will be trace amounts of decolorizing agent on the cells. And if given enough time, the decolorizing agent will remove the color from the gram-positive cells. You then wash with cold water and then flood the slide with safranin. I think we already did the colorizing agent. So wash the slide immediately with cold water to wash off the decolorizing agent. Then flood the slide with safranin. You can apply the safranin to 1 to 2 minutes. Safranin is a counter stain. It's a different color than the primary stain. And it's the second dye we used, and that's why it's called the counter stain. It will stain the gram-negative cells, which are clear, pinkish, so the cells will be easier to view. And then we wash the slide in cold water, getting off the majority of the safranin. You then dot it dry with bibulous paper. And now you can observe the specimen underneath the microscope. Any question about that overall procedure? All right. Let's go on here and then I'll stop. First you stain with crystal violet. The cells, all the cells, will stain bluish purple. You then add the grams iodine after the water wash, which isn't shown here. Add the grams iodine. The cells will be a little more intense in their color, and they'll be a little more purple color than the bluish purple of the crystal violet alone. And that's because the iodine will complex with the crystal violet, making a larger molecule in the cell. You then do the cold water wash, and then you do the decolorizing step that will pull the crystal violet iodine complex out of the gram-negative cells so that they will be clear now. But if done correctly, it will do that, and it will not pull the crystal violet iodine complex out of the gram-positive cells. So this is assuming we're applying the decolorization agent for the correct amount of time. And then you wash the slide immediately with cold water, and then remove the water, and then add the crystal violet, the safranin. And then the safranin will get in the cells and stain the gram-negative cells pinkish, but the gram-positive cells will retain their purple color. Any question about any of that? All right, there's no questions. I'm going to end here. We'll stop a little bit early because I think I've only got a few slides left. Oh, a fair amount, but we're going to stop here. Any questions? If not, I'll see you at 6 if you're in the second lab. I'll next lab if not in the second lab. All
Thank
right,
you.
bye.